My name is Bill. I am the GDC User Services Lead, and today I'd like to talk to you about GDC documentation and publications. So this webinar is going to be a little different from webinars we've had in the past, in that you might think it's kind of odd that we're having a webinar, a support webinar that is on support. And a lot of this stuff is self-explanatory, but I do want to bring attention to a lot of features that the GDC has in the way of support. The GDC support system is pretty big, so there might have been some features that users aren't aware of. So the goal of this webinar is to make you aware of those. The two main sections of this webinar are going to be an overview of the GDC website with some highlights below on GDC support and how to gain access to controlled data, and the GDC documentation website. I'll probably be referring to that from here on as the doc site. And we're going to go over some features of that, including the bioinformatics documentation. So first we're going to go over a little bit of an overview. There aren't that many slides in this presentation because most of it's going to be a demo of the actual documentation site and other website. But here I just want to show you how the GDC support is organized. There are two main branches of GDC support. The first being the GDC website. This contains a lot of higher level information about how the GDC works, how the GDC is organized, and what kind of policies are put into place. And this is also the website that contains the publication pages, which we're going to go into later in the presentation. That website can be found at gdc.cancer.gov as seen below. Within the website, there's a section on support, and that can be used to reach the GDC help desk. This involves a support desk via email, and this gives support on the entire GDC as well as support on data submission. So users consuming the data and users that are submitting the data can both receive support from the GDC help desk. The second branch of GDC support is the GDC documentation site, or GDC doc site. This contains a lot more information for people looking to use the GDC. So this contains in-depth tutorials, information on bioinformatic pipelines that goes beyond the surface level, and a lot of example commands that can be used to construct your own queries and do what you're looking to do. And that website can be found at docs.gdc.cancer.gov by my team. And as I said before, we are an email-based help desk. We can typically help with any GDC-related questions. This involves how our bioinformatics pipelines work, how features of the GDC, including the API or data portal, work, and then submission requests that involve how to use the submission portal or requests for different data sets to be released to the GDC. If you do email us, we will get back to you within 24 hours. We do have some limitations that I do want to make users aware of. One of them is that the GDC has expertise on the pipelines that the GDC runs. We don't have enough context to know everything about a lot of about specific experiments done by different research groups. So usually we will be unable to offer scientific recommendations for analyses that are performed downstream of what the GDC provides. We are happy to give as much information as we can about the GDC data that is provided on the GDC portal. This second one is common sense, but the GDC will not allow users to get around any barriers that will prevent you from downloading controlled access. Any support that we give to help download controlled access data will require access through dbGaP, which we're going to talk about how to get later in the presentation. And the GDC doesn't know everything that other organizations might know. So if you have questions about that another organization might be able to answer better, for example, if you ask a question about CBioPortal to the GDC Help Desk, we will recommend that you contact the CBioPortal Help Desk. So now that's really it for the presentation. I'm going to get into the actual demo here. So I'm going to start out with the GDC website. This is at gdc.cancer.gov, and as you can see, it's divided into a bunch of tabs up here. About the data, as I will highlight here, is a bunch of websites that tell you, sorry, it's about the GDC. About the GDC gives information on how the GDC's mission as a whole, how the GDC's policies work, and various high-level things on how the GDC itself works as an organization. The About the Data section has information about the data itself, like it's advertised. So this includes information about how the data is organized in the GDC with the model and dictionary, what kind of data is offered by the GDC, and this is where the publication page website lives. The Analyze Data section gives information on how data can be analyzed from the GDC and within the GDC. So this includes information on the GDC Exploration page, which contains all of the index mutations, and Community Tools, which is a repository of external tools. I'll show you more about that in a bit. Getting controlled access can be sometimes complicated. So we have a whole section on how to access the data. So this includes the policies regarding controlled access and how to obtain access to controlled data. Submit data, also self-explanatory, contains information about how submitters can provide data to the GDC. That also includes prospective submitters. So a group looking to submit data would apply through a page on this website, specifically the GDC Data Submission Request Form. For developers includes a lot of information about the GDC backend and how that is organized and maintained. Support has lots of user guides and information on how to get in contact with the GDC. And then we have a news tab for news about the GDC and a link to the Twitter. So please follow the Twitter if you would like. So next, because these tabs don't always translate in a friendly way to a Zoom recording, I am going to go down to this part with the sitemap to show you some sections that you may not be aware of that are useful for a lot of users. So first, I would like to show you the page on data analysis processes and tools. This section has some pretty high-level information on how the GDC Dave analyses can be done. And there is also a section on GDC data processing, which has high-level information about how data that is submitted to the GDC can be or is processed in our pipelines. So genomic data processing shows some very basic flowcharts on how the data is processed. For example, It's a very surface level thing on how DNA-seq works. So BAM files are produced, which go through variant calling, producing VCFs. VCFs are aggregated that produce, and this produces math files. We also have some very high-level information about RNA-seq, microRNA-seq, and copy number variation. So if you're just looking to know what the GDC does in terms of bioinformatics, this is a good place to start. The documentation site has many more details, which I will show you later in this presentation. Speaking of bioinformatic analyses, I'll go to the bottom here to browse. Sorry, the link isn't down there. So GDC reference files is a page that can be found under About the Data, GDC reference files. This contains the static files that are used to perform GDC bioinformatic processing. For example, kind of one of the main features here is the GRCH38 reference sequence. This is the FASTA file that is used for alignment in the GDC, and it can be downloaded here. This also includes a lot of other files, including index files for alignments and star, annotation files that includes the GDC data model, and then lots of miscellaneous files for different analyses that might be requested from external users. If the file has a link, that means it's open access and can be downloaded by clicking on the link. But some of these reference files are controlled access. This includes the panel of normal files, which are used for variant calling. These are used when we run Mutect2, and these are giant VCFs that are a panel of normals and require controlled access to download. So these can be downloaded by copying this UUID and using the GDC data transfer tool to download. So the next part that I would like to show you is the GDC data model. Now this can be used by two different groups of people. One, if you are somebody who is downloading and using the GDC, the data model reflects the actual analyses and downstream files that are produced by the GDC. So by reading the data model, you can get kind of an idea of what the GDC has to offer and how these files are related to each other. More importantly, though, if you're a submitter, you can use the GDC data model to guide the organization of your submission. For example, if I needed to submit a bunch of aliquots here, I would be able to see, based on this model, that I would first need to upload samples and cases to a previously established project. And if I didn't know how to submit a type of clinical data, for example, I have lots of treatment data for a patient, I would then know that the treatment here is attached to diagnosis. I know it's kind of small on the screen, but I just kind of want to get across that the GDC data model can be used to guide submission. So feel free to take a look at this page if you would like. Okay. One more thing that I would like to show under Access Data is the GDC Data Transfer Tool. So the GDC Data Transfer Tool, as you know, is a tool that is used to download larger files that perhaps couldn't be downloaded directly through your browser. As you probably know, if you were to download a 50 gigabyte BAM file, it's not a good idea to do that through a browser, since that can fail and cut off your connection. So the GDC Data Transfer Tool, here's where it can be downloaded. We have a description and some system recommendation. The latest distribution will always be here. It's available out of the box for Windows, Ubuntu, and Mac OS X. It can also be built from the GitHub repository for any other operating system that you would like. If you're not familiar how to do this, contact the GDC Help Desk and we'll be happy to help you. In the GitHub repository, you can also download previous versions if you wanted to for some reason. Now, within the Access Data tab here, we have a section called the GDC Community Tools. The GDC Community Tools here are tools that were developed outside of the normal GDC development process. So this can include tools that are made available by members of the GDC. For example, a previous member of the bioinformatics group developed a regroup tool that automatically pulls regroup information from BAM files. And completely external groups will sometimes develop tools that relate to data within the GDC. So this is, here is a package that annotates DNA methylation array data and applies to the data that is hosted by the GDC. These can all be filtered. These can all be filtered by different topics. So something like data processing can be selected. They can be filtered by year. And then they can be filtered by keywords, which will filter these down according to the description, title, according to the description and title. The next section that I would like to show you is the GDC Publication Page. Now, the GDC Publication Page is a section where publications that were generated based on data that is hosted at the GDC can be introduced. So these pages will introduce what the study was about using the abstract. It will give links to access the manuscript for the study, and it can also be used to host files that were generated during the publication of the study that don't fit into the GDC data model. For example, a project might generate a lot of molecular data that is submitted to the GDC through the normal means, and that the molecular data will be harmonized. However, there are other files, for those of you who are familiar, during a publication that are generated that cannot be harmonized with the GDC and don't fit into the data model. For example, a table with some metadata about patients might be a good file to put in the publication page. So one of the examples here, let's use, it's the same filtering method as the community tools. Let's select TCGA as the program. And this can be, each publication page can be accessed by clicking View Publication Information and Supplementary Files. As I said before, we have the abstract, we have a link to the actual publication, and here are the files. In this publication, there are open access files and controlled access files. Now, the open access files can be downloaded directly through these links. So if the file has a link, that means it's open access and anyone can download it. However, the controlled access data may be listed here, but will not contain a link. For example, this file right here would require the manifest to download. So one could go to the GDC manifests, go to controlled access data, download this manifest, and then pass it to the GDC transfer tool that was mentioned previously. The GDC publication page, and I'll unfilter this real quick, also has these groupings of publications that correspond to larger programs that have to do with cancer research. So if you've heard of the Target program before, all of the kidney project publications are laid out in this section. So if you were looking at pediatric kidney cancer, you might peruse these publications here to search for relevant files. And TCGA, you may have heard of the PanCan Atlas, which involves all of the TCGA cohorts being analyzed together. And this contains 22 publications from that program. So that is all I have to say about the publication pages. The next part of this is going to be an introduction on how to obtain access to controlled data. Now, I do want to give a caveat here in that obtaining access to controlled data in the GDC is done mostly with other organizations, such as ERA Commons and dbGaP. So I can go through the basic steps used to obtain controlled access data, but specific questions would have to be directed to the specific organization. So here's the basic walkthrough, and I'm going to go through the steps now. So the first step is to obtain an ERA Commons account. This is the account that is used to log into the GDC. So if you were to go to portal.gdc.cancer.gov, you would see a login button here. This is where the ERA Commons login would be performed. So you would log in here using your ERA Commons account. So using that ERA Commons account, which can be used to log in not only to the GDC, but to dbGaP, you would obtain access to dbGaP through their system. Once you did have dbGaP access, you would go into dbGaP and gain access for a specific research project. So one question you may have is, how do I know which data set in dbGaP corresponds to the data set in the GDC? And my answer for that is the PHS ID. So if you were to go to projects, and let's say hypothetically you wanted access to this project here, FMAD, which stands for Foundation Medicine Adult Dataset. You could go to the project page, click on that project, and you would find a dbGaP study accession here. This is the PHS ID that I was referring to before, and this can be plugged into dbGaP. So back to the controlled access, you would log into dbGaP, as I showed before. You would create a research project. This brings you through kind of a user interface that you fill out different fields. It's pretty self-explanatory. And when you get to the part where you are requesting specific data sets, you would enter the study accession in the study accession field. So I'm using study accession and PHS ID as synonyms. Once you got that ID, you could specifically query a data set and gain access to that, or request access to that through dbGaP. It does require that you describe your project, give information about your institution, that kind of information. Your computational resources and how this data will be stored is important too. So once you do have access, there are options to grant access to other individuals in a lab through the dbGaP website. The GDC doesn't have an overarching policy about this. I think it is specific to certain projects. So if you do want to grant access for other individuals, I would suggest reaching out to dbGaP for information about a specific project. So once you have all that and you do gain access, you would log into the data portal like I showed before by clicking on login, and automatically it would show you which projects you have access to. So that is all I have for gdc.cancer.gov. So my next step would be to move to the documentation site, which is docs.gdc.cancer.gov. So, like I said before, the documentation site contains information, specific information on how to accomplish certain things in the GDC. So basically, if you want to do something in the GDC, you would click on one of these tabs based on what you were looking to do. So the API here the API tab has information on using the GDC API. This includes things like querying for a certain data set, running some of the data analysis endpoints, BAM slicing, and submitting directly through the API. The data portal contains information about, as it says, the data portal, how to use it, what features are included, and how to use those features. Data submission has not only information about the GDC data portal, but information on how that portal can be used and how the submission system itself works. The GDC data transfer tool section, like it says, has information on how to use and troubleshoot the GDC data transfer tool. Data dictionary has information about the GDC data dictionary, which is how we maintain our data quality at the GDC, and this gates submission and allows only certain things to be submitted to the GDC. The data section has information about the GDC data model, different file formats that are offered at the GDC, and then specific information about multiple bioinformatics pipelines. Lastly, the encyclopedia here is, like it says, an encyclopedia that gives small articles about a lot of terms that you may hear while using the GDC. So the first feature that I would like to show is the API documentation. If you're looking to search for information within the GDC, the search and retrieval place is the best website to start at. This website includes the components of a request, so how to perform an actual API request. These are the specific things that can be queried. We have specific command and expected output features here. So if you were to put in this command, you would expect an output that looks similar to this. Note that sometimes the order can change in these outputs, so they won't always be 100% the same, but you will find something similar when using our test shell commands. There, the page basically goes on about other more in-depth features of the GDC API. So for example, file version information. Next, I would like to show you a page that we put together because the GDC API can be accessed using curl or a web browser, but it can also be accessed using Python. We wanted to put a few examples of that here. Now, each of the Python scripts here are hid behind a tab system here, so the page doesn't get too crowded. But for example, if you wanted to see how to do a basic query using Python, you would click the Python script here, and this this script would be shown. Most of these scripts require installation of the requests package because that's what we use to connect to the API. And I think most of these were developed using Python 2.7, but I know most of them do work with Python 3. So they should work with Python 3, but let us know if they don't, and we will definitely update them. Not only are there basic queries, but there's some things that are a bit more complicated. For example, how to download a controlled access file and how to pass your token to the script to download the file. So here's how to download an actual file. And if I didn't show you before, here are the links to download the actual script rather than copying and pasting out of this window here, as Python can be quite touchy with indents, and copy and paste doesn't always go well with that. Okay, so the next section that I would like to go over are some of the sections that describe bioinformatics pipelines in the GDC. First is the DNA-seq analysis pipeline. This is how we process whole exome and whole genome sequencing. Now, first, we kind of give a high-level description of how the data processing steps happen. So these are just paragraph descriptions on how the alignment works, and some basic flowcharts that show the whole process. For those of you who do want to repeat this, whether to run these analysis of your own or to check our work, the shell commands that are used with their corresponding parameters are laid out here for each of the steps. So, for example, this is the step used to convert submitted BAM files to FASTQ so that they can be realigned using the GDC's parameters. BWA alignment is included, as well as some conditional pipelines. So, for example, if the mean read length is less than 70 base paired, BWA mem wouldn't be run, but BWA align would be run. So as you can see here, there are lots of information about how the DNA-seq pipeline is run. Usually, and you can navigate using this side panel here. Information about variants that aren't performed through the same harmonization pipeline as most of the GDC are described here. So for example, Gini variants are performed using a specific liftover pipeline, and this is described here. At the bottom, we have the file access and availability, which shows which data types are produced and which file format they're made available in. Now, the next part I want to show you is the mRNA analysis page. So this is basically the same type of page as the DNA-seq analysis pipeline. As you can see, there are flowcharts, the same type of information, input-output tables, but the GDC switched our RNA-seq pipeline about a year ago, and so we document both of the pipelines here using this tab system. So if you want information on RNA-seq alignment from data that is before data release 20 or data release 15, you can see it here. If you're looking at data that was released with data release 15 or afterwards, you would see the command line parameters here. The same thing is done for mRNA quantification, and then we have some additional information about how our quantification works, specifically how FPKM and FPKM-UQ are calculated. So lastly, in the bioinformatics section, I'd like to go over the file format MAF section. So GDC MAFs are produced using a specific format, and this format is detailed on this page. So information about how the somatic MAF file is generated is first presented here. This is a list that tells what the GDC did verbally, but as you can see, we have a flowchart here that shows a variant coming in from a protected MAF. If it meets certain conditions, it can make its way to the public open access somatic MAF. So all these steps are they're a bit bare bones in this flowchart, but they're detailed a lot better up here in this section. math files are, as you are aware, are large TSV files, tab separated values. They have more than 100 columns and minimal column information. So here, each of the columns are detailed based on their number and their title, which are put in this first column here and described in the second column. So as you can see, there are a lot. I'm not going to go through all 126 of them, but if you are curious if a certain piece of information is in our math files, or if you found information in our math files that you weren't sure how to interpret, you can use this guide. Lastly, the impact categories are laid out here, because within the math, certain mutations are assigned impact categories based on their hypothesized effects on a gene. So these 3 different categories are assigned based on 3 different algorithms that includes VEP, polyphen, and SIFT, and each of these has different vocabularies, but all of the vocabularies are defined here. So that is the last thing that I wanted to say about bioinformatics. And because we have a little bit more time, I'm going to very quickly go over information about the data dictionary. So the data dictionary viewer, which can be reached by going to data dictionary and viewer, kind of lays out in a table form all of the different entities that can be submitted to the GDC. So if I were to click sample, I would see all of the fields that can be submitted to a sample and the allowed values for each of those fields. Additionally, if you want to know whether a field is required or not, look no further than this column here, which will say yes or no. There's also a link to the external database that that curates these specific properties. Kind of on a to expand onto the viewer is the data dictionary search tool. So here it is. If you're interested in submitting about a specific type of cancer, for example, lung, this will list out all of the properties that have information that matches the lung term. So for tumor codes, a lung cancer is available, which matches to a specific tumor code. So under the sample, there is a field called the biospecimen anatomic site in which lung can be entered. And there are many other different ways to get lung cancer across in a data submission. If you were wondering about certain properties, for example, tobacco use, so let's say you had a bunch of data on patients' tobacco use, you would enter tobacco into the data dictionary field, click on properties here, and you can see all of the data fields that have information on tobacco use. So if you had a list of patients and what type of tobacco they used, you might want to check how that can be entered. So you would just click on see all values, and a list of all the acceptable values from the GDC will pop up. And also show you other terms that may correspond to these values. So if you were to have e-cigarette in your data, you would see here that obviously corresponds to electronic cigarette. Not all of them are that obvious though, so this is why it can be helpful. Lastly, the dictionary term here is just kind of a giant, just lots. It's everything is listed out in different categories, and each of these can be expanded to see how all of these relate to each other. So if we collapsed all of them, we would see that anything related to tobacco is limited to the exposure entity and the follow-up entity. And looking under the follow-up entity, we would see that everything falls under the risk factor property. Okay, that is all I have for this webinar. Here are some helpful links to help you get around the GDC. I've highlighted the dictionary viewer and dictionary search in case you wanted to play with that. But the GDC website and documentation site are the two most important resources, as well as the GDC support email, which can be found here. Thank you so much for listening and for attending this webinar. I'm happy to take any questions that you have now.
Thank you, Bill. So we have one question. I was wondering if this recording will be made available after the event.
Yes, this recording will be made available. It sometimes takes us a little while because all of the recording needs to be subtitled, and it needs to be edited so that it flows nicely on YouTube. But this will be made available. All webinars will be.
Thanks. If there's another question, please put it in the Q&A. Okay, seems like that's the only question today, Bill. Okay, so.
Great. Okay, well, thank you everyone so much for listening, and you all have a great rest of the day.
Bill, you want to share the next seminar we'll have?
Oh my goodness, I do want to share that. Thank you for the reminder. There will be another webinar coming next month on July 27th at 2 p.m. Eastern about the GDC RNA-seq data processing pipeline. So this will be an in-depth look of how the GDC processes RNA-seq data, and this will be a collaboration between me and the bioinformatics team.
Okay, great. Thank you. Thank you everyone for attending. Thank you, Bill, for the seminar.